That's sharp knives there.
Nice grab.
It's also branching.
That's cool. So Quinlan, when you guys are sampling this, do you guys just keep it? How do you preserve it?
A good question. So when it arrives, when it comes
All right,
out of
stick
the
in the
water
box.
to the surface,
Starboard
we
box?
have to carefully place it into a container where we have to take photos of it. And
You
then we
can,
have to give
zoom
it
back in, Amber.
its ID and of course take photos. And then after we store it in ethyl alcohol 95%.
And some of the bigger specimens get dried as well.
Oh, that's what I was gonna ask. Like, so I'm just used to botany on land, so I'm like, do you plant press this or how do you preserve it?
No, if we dry it, we just like hang it and let it dry.
With a
Should be good for
subsample
full
for
zoom.
genetics.
Yeah,
usually.
if we do, yeah, absolutely. We do always keep some in ethanol for genetics.
Yeah, that makes sense.
You can open the box for me.
I'll help you.
So I guess let's just log this as unknown Chrysogorgidae
on
because
the,
I'm clueless.
Turn on the light, please.
Copy.
Yes, this is exactly why we make these collections, because we encounter things like this that sort of defy the expectations of even some of the world's experts. This coral's quite large.
You can go wide now, Amber.
Shape and
I'm
branching
gonna dead
pattern
stick
that
here while I put this in the
is
box.
somewhat familiar, but not quite. So this could be new. This could be a different growth form of something we are aware of. And you can't really tell from video if it's new.
Why? Okay, that's completely dead stick. I'm just gonna let it
Are
float.
these rocks basalt?
Most likely, yes. Yeah, manganese encrusted basalt is most likely.
We're gonna float up pretty quick, so you're gonna have to come up there as well, Jaden.
Copy.
It's super light coral here, so I don't want it to blow away.
If you need to stick it in one of the bigger boxes,
Yeah,
that's fine.
Can I put it with the rock?
No.
Roger. Keep coming up, Jaden. Come up a little faster. I'm floating up like crazy here.
Copy.
Okay, you can close the box. All right, can close it. What we're doing, I just turned off all the thrusters and we're pretty late, so floating up rapidly. I didn't want to blow it away. It's so wafty. Okay, I'll come back down now.
So Dan, how much space do we have for samples?
We have sorry, stand by.
Quinlan, looks like you got a typo in the sample number.
Oh. Oops, my bad.
No worries.
Yeah, sorry, that was supposed to be sample number 17 in starboard box F.
So if you want to just in C log, just make a little comment and correct it.
Okay, Chris, I think you can start moving on missile again.
Roger. Bridge now, 40 meters, 250.
And we do not have the tether turn, like magic. You can bring your head to 250, Jaden, that'd be great.
Copy.
Sorry, when we're sampling, there's a lot of, we actually have to pay a bit more attention up here because we get focused on the sample and then, you know, Atalanta runs over us, manage the tether, manage the ship. We tend to have our hands kind of full there. Manipulator moving and the cameras. Sample boxes. We have 2 sample boxes. So the front one has 2 partitions and the back one has 2 large and 4 small. I used to know the cubic measurements. Roughly in imperial units. The front one is about the size of a small. The larger ones are the size of a small fish tank. We actually have what we call fish tanks that go in them. So the scientists can just then pick the fish tank up out of the sample box.
Dan, do you want the starboard port lights off again?
Yes, please. Thank you. And then there's, I think, what, 7 jars on there, including the flush jar. So we have 6.
Including the flush jar, I think it's 8.
Maybe it's 8, yeah. Which they're about the size of a, I don't know, maybe a 1 gallon, 2 liter container, something like that. You know what we haven't done is any push cores. It's a perfect push core territory too.
Yeah, there's also a reason for that.
Yeah. Nobody wants push cores, is that why?
No one on this boat.
Mud people will be so disappointed.
The catch with the push cores is largely they're only good in, or they're most valuable in replicate. And so just taking the occasional 1 or 2 is a lot of processing and preservation time for limited value. You kind of have to do them in replicates.
Yeah, an array of them.
Got some very noticeable straight line feeding traces here. You can kind of see these tracks running in the seafloor, and that is the path of a sediment-eating organism. And normally they're kind of windy and, you know, like if you sit there and watch a snail for an hour or 2, which I'm sure we've all done, and how they kind of wander back and forth. These look like this, whatever made these had a purpose in mind and kind of went in one direction.
Here's one potential culprit. It's these sea cucumbers.
That's why. What's up with the DVL?
Huh.
That's why it was being so wonky. Huh. That's weird. Yeah, I'm going downhill, that's why. So the back was too low.
So good morning from Palau. It is 6:19 a.m. here in Palau. We are currently at 2,057 meters. This is our 4 to 8 watch team. So I highly encourage our viewers to Check out this talented team's bios on nautiluslive.org.
Bridge nav, another step.
Can push in just a little there, Amber. We're going downhill, so let's see the seabed a little better there.
Yeah, so we landed on kind of the local high on this
One more step, please.
this area that to me in the multi-beam looks like a landslide sediment pile, and we're moving kind of off the sediment pile, do a little valley, and then I suspect we'll find hard grounds, or more likely to find hard grounds once we get across this little saddle and get on the seamount proper. We wanted to come out here for Harold so we could maybe find some fish. But
So is that what you guys are really hoping for, is the fish?
right here, yeah,
Not seeing a lot.
but not seeing a lot.
Yeah.
We're on,
So we tend to see different fish depending on the seafloor habitat we're in. Some fish like to pick things up from the sediment. Others pick up invertebrates from the hard substrate, maybe even the corals.
Any fish in particular you're looking for right now, or hoping for this dive?
Well, we're interested in the complete biodiversity of the fish community, so we welcome any fish that comes into the frame.
they like rat tails, don't lie. They like rat
Well,
tails.
Jeff's favorite is the rat tail, the macruids, and these are a fairly common scavenging fish. So back in March, Jeff and I, another student from our lab, Aaron, were out here with National Geographic Pristine
Speaking
Seas.
of fish,
Oh, there's fish.
Beetlejuice,
There's a fish.
top right.
Oh, this looks like a cuskeel. So we came out here with baited cameras and looked at all of the scavenging and mobile fauna around Palau.
Come down 5 meters.
So we've been delighted to see some of this familiar faces from that survey throughout this cruise. Getting some much better shots of them with our fancy Zeus camera. That's it. Come closer. Thank you.
Good fish.
Oh yeah, and if we could get the lasers to cross right over it, please. Swim.
Oh, there it goes.
Oh. Okay. It's good enough.
There we go.
Got any more zoom?
I think I'll push out a little, Lambert. That's good.
Great.
Photogenic.
Oh, it's even in the still cam. Okay. I think we're good. Thank you.
And we're going that way anyway, so.
Oh. Wow, just really showing off here. Oh, see ya.
So this watch team will return back 4 p.m. 4 p.m. to 8 p.m. Palau time.
Bridge nav, another 40 meters.
Hard bottom.
Can you fix the nav screen boxes, please? Thank you. Come down 5 meters, Jed.
Copy.
I know one of our viewers said our watch team should be called the Oops Alliance, respectively.
The Oops Alliance. Wow.
Oops.
Oops, wrong button.
Backseat drivers in the chat. Wow.
I like it. Earned it. Sorry, Amber. Should have warned you.
All good.
Okay, push in there.
Copy.
It's good, good. Thanks.
This is another Chrysogorgid coral. With a squat lobster.
Do you want a tight zoom or?
Yes, please.
Okay, go ahead.
Europtychus squat lobster associate. That's about all I see. You can kind of see why this family is called gold corals, how gold in the skeleton appears. Right, science is happy, thank you.
Roger. Moving on.
We got some nice botryoidal texture on the rocks.
Now what Herc does when it finds a cliff,
Ooh.
it runs out as far as it can and jumps.
Base jumping Herculeses.
Coral or
You'll
sponge?
have to come down a good
It's looking like a
5
sponge.
meters for me.
Copy.
Shout out from ONC, Monica. Hello.
Hello.
So this is a large glass sponge with a coral at its base. Oh, there's another epic coral in the background too. Ooh.
Where are you in the DSC? Is it below me?
It is still below. You need to come down.
Roger.
Keep coming, keep coming. You're just in frame now.
Right.
Okay.
It's a big sponge.
Picture
It is a
time.
quite large sponge.
Can we turn the lasers off?
Copy.
Thank you.
Great. Harold, you happy with still cam shots?
One more. Yep, I think we're good.
All right, down bottom right is a really significantly sized coral. When you're done with the pretty pirouette, let's go down that way.
Bottom right.
It's gonna be now lateral right to you and down probably another 5 meters,
Right.
but I think it's worth the effort.
Down to the right.
That's what it is.
So the sponge is a Tretopleura, and they have this really interesting coral-like morphology. Sponges and deep sea corals are both filter feeders.
Seen it in the,
No, I'm not. You might want to, it might be under the vehicle at this point since you
Right.
pirouetted over. It's tall, so don't come down too much.
right.
So is it very common for the sponges to grow horizontal like that?
It's not, yeah, it's not, it's, so sorry, if you look in WCA upper, it's just under the craft arm.
15070.
Yeah, there doesn't seem to be a lot of, something that I'm pretty interested in from a research perspective is how the current flow kind of moves and how filter feeders get on different angle surfaces and how it reflects the current. But yeah, it's not that uncommon to see them off the side of shear walls like that.
Wow. That is cool.
All right, Les, it's a bamboo, so I'm looking at you.
Yeah, this looks like Tridentices candelabrum. That's my guess.
Yeah, that would be mine as well. But when Les is when Les is in chat, I don't identify bamboo corals. Can you come down just a smidge more and get the whole thing in Thurber?
We're on the seabed dirt.
Oh, okay.
I could back up a bit,
No,
but then I'll
yeah, no,
wreck
it's not
it.
back up.
Yeah, you know what happens when ROVs back up.
That's so cool.
You're steady, so take the shutter speed to like 1 and 125. Drop the f-stop to like 7. And now drop the ISO to like 2000. You can drop the f-stop even more. Take it all the way down to 5.6 or 4.5.
Roger.
Now focus.
Yep, happy.
Now you have to manually focus it. All right, can we get a tight zoom on HD?
Yeah, go ahead. Hammer.
Copy.
So Les Watling, who is one of the world's foremost experts on this group of corals, is saying that there's a big gap in the distribution of this species from the Central Pacific to the Great Barrier Reef, and this is the first one that's ever been seen in between them.
I believe it's not the first one we've seen on the expedition, though. This is
That may be
certainly the most spectacular.
This is a great example of a nodal brancher. You can see every branch is right at a black line, so it's branching right at the nodes. Can we just pan and tilt around a little bit and look and see if there's any associates?
Where do you want to look?
Just around. Doesn't.
Right. Where's the one branch there? It's got a. Sorry, let me slow that camera down a little bit.
Yeah, it looks like a break or something, or just a big bend.
Yeah, there's one.
Yeah, I see it. I don't have an answer for you.
Roger. See if I can do the Jaden super slow.
One of the biggest ever seen, according to Les.
Let's get a good shot of the base too, please.
Roger.
What's the estimate height for this one?
It's tall. I don't have a
I missed
better
the
estimate
laser.
for you yet.
Yeah.
Great. All right, if we can get one more whole colony shot with the lasers on, I think we shall be done.
Roger.
Yeah, somewhere in the 1.5 to 2.5 meter range. Okay.
Early results indicate it's big.
Yeah. All right, we're done. Thank you.
Roger.
Les here pointing out that the name is a bit of a misnomer because if you look at the base, it's not actually a trident. It's just a Y shape on this particular colony.
If you're going to do a pirouette, you want to drop the lasers.
Yeah, go ahead.
really interesting curvature.
Yeah, mesmerizing.
Didn't really notice that from the head-on view.
It's like a rake.
Yeah.
That's exactly what it's doing.
I know I have a sense of the hill here, but might be able to get a reverse DSC if you want. So I'll come down and what happens. Come down, 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 down. Yeah, I know which way the current's going now. Trying to push me into it. It's quite a good breeze there from behind me.
It's not the direction I would have expected looking at the morphology of the coral.
It might be tether, not current, because I was looking at Atalanta and it's probably what it was. I turned and put the tether over my head, so it's the tether effect.
Swimming isopod.
Yeah, I was mistaken there. I let go of it. It's Hercules is not really going anywhere.
Okay.
The particulates raining kind of down, so.
Yeah, the bed forms wouldn't indicate a very strong current here.
Would indicate a strong current?
Would not.
Yeah.
It's like another one of those strange Chrysogorgias Chrysogorgids we sampled already.
Okay, I need to turn this way for tether management. Completely lost the plot. Which way are we going? Back the way we came? That's right, I jumped off the cliff and turned around.
One of the things that fascinates me about coral and sponge distribution is we've got these two exceedingly large, super healthy colonies out here. So obviously there's food availability and current flow, but we've only got two. I don't have a good explanation for why there isn't more here, or why we don't see different size classes or different age groups or anything.
For a minute, and then we'll jump off.
These just two really big colonies all by themselves out here. So obviously the conditions are good enough for these individuals. I don't know why you don't have more.
Okay, turn Turn and burn. Gonna come right under the other grid. Back down into the abyss.
Yeah, and Les is pointing out that one of the factors that probably controls those is larval supply, which absolutely makes sense.
Fish. Looked like another ophidiid, maybe? Cuskeel?
That
Didn't
was
get a
my
good
guess.
look.
Yep. Fly through my own dust cloud here. He said stand by. Okay, Jane, you can turn to the 250 heading there. You'll have to come down.
Copy.
When you get tether, can we look at that, please?
Roger.
Going back to the bamboo coral lake. How would you estimate the age of that?
I think it's, if you were to take a branch of it, they've got growth rings like a tree.
Oh, okay. That's super cool.
And you can also do some radiometric dating on the skeletons.
And come down another 5 meters for me.
Copy.
Okay, Emery, you can zoom there. That's so far away downhill, but let's see what we got there.
Anthropitherian.
Bouncing low there on the tether, sorry.
It's okay. Found, you see how it found one little rock out here in the sediment to grow on. All right, that's good, thanks.
Okay.
Copy.
Good one.
So what happens if it outgrows its rock?
It falls over.
Okay.
And then it's done. Or
It's not, it's not definitively done, but it's going to be a lot harder for it to survive. And if it, loses that 6 to 8, 10 inches of height into the water column. So yeah, it would probably slowly starve.
Do they kind of self-limit in a way, or just keep growing?
I don't know. I don't think I certainly don't know. I don't think we know. I would anticipate or guess that no, that
Yeah.
they probably don't have any feedback mechanism to know stable their rock is. And in places like canyons and stuff where you do get a lot of, rock sloughing off and stuff like that, you do see some really big colonies that fall down, clearly. Like they break off. He can't reach that far. We're stuck.
We have seen coral fans that have been toppled over some time ago, and despite the fan being just lying flat on the seafloor, the new branches are growing upward.
Yep.
That's amazing.
So it's not like a plant or a tree where it needs to be rooted to survive. As long as it gets food to the polyps, it'll keep growing.
As you get some tether, there's a fish out of frame to the right, but I don't think you can get there yet.
All right.
Oh, cool. I have a shout out, my hometown, Navajo Middle School.
Very nice.
7th grade class watching us right now.
Hello to all of you. If you have any other questions, send them our way.
Yeah, hello, hello.
Okay, good for a half zoom there, Amber.
Copy.
You can try and go a little tighter. Downhill again, so.
This is a rat tail of some kind. It's got interesting coloration on the tail. Looks almost transparent or white.
There's some xenophyrophores in the sediment too.
Oh. I'm guessing it's a Coryphonoides rat tail of some kind.
That downlight some.
Yeah, they're off.
Turn them on.
Copy.
Okay, you good?
Yeah, it's
Yep.
a good zoom.
Thanks.
Do you want the downlights back off?
You can leave them on for now. Let's see.
Copy.
Here's another variety of sea cucumber.
Looks like it's a blue one.
Yep.
one's very broad looking.
Zoom coral, please.
All right. Go ahead, Amber.
That's pretty.
Yeah, this is a bubblegum coral.
Bubblegum coral.
Yep, or paragorgid with a big astroschema sea star, or sea star, brittle star, living in it. All right, thank you. Zoom there, please.
All right.
Oh wow, I didn't even see that tall one right there. That thin,
Okay, Amber.
that strange Chrysogorgidae is really hard to see.
Push it a bit more, thanks.
A couple more of these kind of bushy Chrysogorgias we looked at earlier, or similar to. However, we may want to stop the ship if it's moving. Need a close look at that, please.
Yeah, what could that be?
Push in there. Amber.
Copy.
Come down, Jaden.
Copy.
Can't tell if it's an anemone or some kind of egg.
Oops. Sorry. A little fast on the ups.
Huh.
Check there. You can give me a few more meters.
Can we spin around and look at this coral from the other from the other side?
Yeah.
Looks a little bit like a mollusk egg, but I don't want to insert my bias towards eggs.
Push in a little for me past the porch there.
Copy.
Okay, can rotate
Okay.
there for him.
Go back.
Huh.
Yeah, it's an egg.
What type of egg?
We really don't know.
Hmm.
First guess is some kind of octopus, but we don't know.
Yeah, I don't really recognize this. Could we potentially do a collection?
Sure.
We're probably going to need to take the whole colony, pretty much. In order, if we're going to do it, we'll get the squat lobster as well.
You can come down a few more meters there. You got 23 under you, so you come down the par. You might have to spin your clockwise 180.
Copy.
Yep. We have some agreement in the chat that this could be an octopus egg. And from the baited camera survey,
Yeah?
we did see one octopus.
That's where I saw it.
But it was quite a bit shallower.
Yeah. There's Atlanta's aft camera too. We're right on the deck with Atlanta.
So this would be quite interesting to sequence and find out.
Do you have enough imagery of it there?
These types
Yep,
of samples are of particularly high value in general, but for my
You can
personal
stay tight
research
there, Amber.
interests, it's good to know what animals are using
So
deep sea
full
corals
zoom, is
as
it?
a nursery
That's full
habitat
zoom.
like this,
Roger.
so that we can figure out what exactly the functions of these animals are that they're performing for the overall ecosystem. So we need to protect the corals if we want to protect whatever's laying their eggs on them. But we have to figure out who's laying the eggs.
The boat still moving, is it? Or it's just stopping there? Okay, Jaden, keep an eye on that tether there. You're gonna probably, when you do come up, just come up
Very interesting.
super
Les
slow.
in the
Or
chat
if you do,
saying that
try and
they
get
collected
this
one similar to this in the New England seamounts, and an octopus emerged from the egg in the bio box.
Wow.
I don't know if we'll be so lucky this time, but maybe we can shine a light through it once we get it in the lab, like we did with the skate egg.
That'd be so neat.
So you want the whole animal, do you?
Pretty much, yeah.
Yeah, I think that's the only way to go. And these squat lobsters are also of high value, since they can tend to be pretty diverse. So this is overall a great sample.
Stay.
Don't run away.
You can just go straight in a forward box with this if you want.
We're at it. Can you rack the HD camera back?
Copy.
Zoom out on the upper camera. You can go wide, Amber.
Copy.
And let me bring the arm up before you open the box there. gentle so the squatty didn't run away. Okay, go ahead on the, open the box for us.
Never mind, the box got in there.
Still got our lobster.
Yep.
At least one. Perfect.
Awesome, thank you.
Amazing.
Okay.
The egg looked buoyant as well.
All right, while we're here, there should be a coral off to our left that I'd like to take a look at before we depart.
All right.
And just as we take these samples, we just want to be very appreciative and thankful of the space that we are in, the Plum people, as they want us to use it for educational purposes.
Yep, that one. This looks like the exact same thing we sampled at the beginning of the dive, but it's kind of strange looking, so I wanted to get a look at a second individual.
Yeah, it's a real privilege to be here and be able to look at these landscapes that nobody's ever seen, and with minimal impact, take what we can for research and try and figure out what's going on in these ecosystems.
Okay, Amber.
Pushing it.
Visible coral there. Pen left just a little for me, Jaden. Well, hold on. Because what's happening here? Sorry, I gotta reposition. The wrong button.
It's a Protoptilum sea pen in the background there.
What that is? I saw it, I just wasn't sure what it was.
Or I think that's what it is.
Good for a push.
Yeah. Sorry, I don't know why Hercules. Always the archer, never the arrow. Blaming the vehicle for doing exactly what I told it to.
All right, thank you.
Okay, all good.
And yeah, I think we can, and if we can just on our way out, fly by the bamboo, you can see an WC upper on the right side.
Roger.
Just want to take a quick moment and wish my husband a happy birthday today. Happy birthday, Lionel.
Happy birthday.
Happy birthday.
And what's my tether turn action like? Dealer's choice, and come around this way. We're going to turn and go the other way, so come around this thing. Pushing just a bit for our sampler as we come around. Okay, I think we want to go this way, right?
Yep.
All right.
255.
255. Okay, off we go.
Ready?
Sure, yeah, by the time you get moving, I'll...
Bridge nav, 50255.
If you bring your head to the left, does that take our turn back down to 0.5?
Copy.
Point 3. Thanks, Oriel.
I'm gonna just stay under you for a while, Jaden, just because we're on the downhill here.
Do you want me to come up?
Yeah. Come up a little bit.
Looks like a dead sponge stalk with some hydroids and maybe barnacles. Another probable Paragorgia, another Chrysogorgia, very similar to the one we just sampled.
Got a little bit of weather today. It's telegraphing the bounce through the tether there when we're tight.
All right, for our viewers, good morning. It is 7:14 Palau time, AM.
Come down a few meters for me.
Oh, this is our second dive for Libu's Voyage 2. We are making our way up. Unnamed seamount. About 70, 80 miles west of Babeldup Island.
And look to your right a little for me. See what's coming up on the left side of Hercules. You're gonna have to come down dangerously low there.
Can you say that again?
Said you can sneak down a couple more meters till you, if you hit the rocks, you're too low. So I'm deeper than you on the cliff face here. Maybe just 10 meters. That should be good there. Bonk. Don't do that. Watch out for that big rock right there. Don't see that one coming. Now you can come up just a little bit, dude. Okay. Look down into your right some more if you can.
Another stocked glass sponge. Is this Calophagus?
That would be my first guess.
You want to see that one up on top there, Brian?
Yeah. Let's look at that.
Yeah, I've heard the story from the New England Seamounts about the octopus egg from Tim, so I think he has some detailed records of it.
Cool. Be curious to learn more. This looks like another sparsely branching bamboo.
You can spin around and look at Herc if you want, and then come down. You're in the deeper water now.
Copy.
You'll probably have to, can you do both? You're going to have to come down. I'm going to drop to the bottom here.
A little Persingid hiding there under the crack. Oh, flyby sea cucumber.
Headless chicken, as we like to call it.
Headless chicken monsters.
Yeah, headless chicken monster, thank you.
It is the name for those in the Gulf of Mexico. Can we zoom there, please?
Sure. Go ahead and push in for a center.
It actually looks like two corals. A black coral in front, and I think my eyes might be playing tricks on me. I think there's a, yep, no, there's
Yeah.
Oh, yeah.
Chrysogorgidae militaris, I think.
Three.
Oh, three, you're right.
Three.
Three. So that's a Chrysogorgidae on the other side, and that might be a little Militaris on the back. And this is, I believe, a Pranopathes in the front. And I have no idea what that swimming thing is. All right, thank you.
You can come down a bit more, good.
Copy.
Thermocam is, at least the controls of it are starting to act a bit weird. Does someone else have control of it right now?
Nope. Not that I know of.
None of my buttons are working. There's like windows everywhere.
Well, give it a second. It's not a very powerful computer. Let it try and catch up. And
Yeah, I probably clicked it too many times.
And the evidence is frozen on the screen for the world to see. I can reboot it if you lose patience there. Well, it's a cool feature, but Atlanta moves on. Which way do I gotta turn? Counterclock. Come down.
cool pillow basalts.
Come down another 5 or 10 there.
Yeah, you can really see the, I don't know what you would call that, the sort of crystallized
Yeah.
shell.
This is a lava formation that only occurs in basically underwater. We get the lava rapidly cooling with the water.
Okay, I'm gonna turn and burn off to the west there. Stay as long as we could. Cool little feature picking up there in Atlantis sonar, where we just came from.
Bridge nav, 50 meters, 255.
Still seeing some bed forms, so there is some current here, but it doesn't look like it. Looks like it's pretty uneven and light. And then what I'm looking at here is just literally the ripples in the sand and the sediment. Can give you a little bit of an idea of the prevailing currents in an area. Any luck with your coming back to life?
Video, do we have a channel for sunset? Or sunrise, sorry?
Not sure.
Oh,
Let's try this.
yeah, I think it, I think it rises at
Can you tilt down?
5:52
a little?
a.m.
Tilt.
Yeah.
Do what, sorry?
Tilt the HD camera down for me.
This is wood?
Is that wood? Yeah, can we look at that?
Yeah, and just spin around to get it on the uphill side here.
Oh, yeah, it's way up now.
Yeah, sunrise is early.
Tilt down a bit.
It's
more,
full
Jade.
daylight outside.
It's going slow.
Still beautiful.
Yeah,
All right. Thermocam is back.
that's good. Tilt down a
Oh,
little
that's
more.
beautiful. Thanks. Oh, yeah. Yeah. Maybe channel 3 next time.
Push in a bit there for us, Amber. Thanks.
Thank you.
That's good. That's good.
So these woodfall communities are pretty unique ecosystems.
Come down a bit, Jade.
You can see all the different life cluster on this, eating this piece of wood, and the biochemistry at play here is pretty complex with a lot of symbiosis, and in order to be able to digest all the cellulose and really recalcitrant wood fibers, but eventually it'll all get eaten. Can we get a tight zoom?
Yep. Is that all you got?
Oh, that's neat.
There's like a little micro crab in each little hole.
Yep.
Yep. All right, I think we can keep going. Thank you.
Roger.
Bridge nav, 50 meters, 260.
Some of those worms are xylophaga. Thanks, Aaron Judah.
Zombie worms?
Is that what they're called?
No, don't think so.
We visit a whale fall at ONC every year. And
Yeah, those are the Osidax.
Yeah, you're thinking about Osidax, Acidex.
I don't know if xylophaga is a common name.
theory it starts going uphill here.
At some point.
Atlanta's picking up something there off its starboard side, but I'm not seeing it.
Fish.
Why do you say fish?
Or shrimp.
Oh, shrimp.
That's a
Sorry,
shrimp.
shrimp.
Aristeid shrimp. Do we have time for a zoom?
Sure, go ahead.
Swimming right into the ground. Looking for the rostrum on the front of the head.
Is it swimming in the ground or is it trying to eat something in the ground?
Oh, it could be. Yeah, not much of a rostrum on this one. All right,
thank
you.
What is the rostrum?
That's another one.
The rostrum is, it's like a little rhinoceros horn on the top of its head. Sometimes that's a feature we use to ID them.
More wood or rock?
Unsure. I don't think it's wood.
Rock.
Almost look like heavily rusted metal. Couple more feeding traces here. You can
those star-shaped patterns.
Yep, Sapunculin holes, brachyurans. So in the center of that kind of star-shaped pattern is a hole, and this worm puts out its feeding appendage and kind of, drags things in and out of its hole. Every once in a while, you can see it as you approach, snatch its arm in, but it's pretty rare. They're very lightly colored, translucent, and they're extremely sensitive to any kind of motion.
Has anyone ever tried to catch one with a push core?
I'm sure, I'd be surprised if one hasn't. I have not been present for that attempt.
That thing dropped out, didn't it?
Could we look at this cucumber?
Sure.
Don't think we've seen this one yet.
Not on this dive, no.
It's an interesting
Okay.
trough in the sediment
It
there.
is. I was just puzzling over that.
A whale, maybe.
I wonder if that's, what do the beaked whale feeding traces look like?
The ones I'm used to are deeper than that,
Go
but
ahead,
I
go ahead,
certainly
Amber.
wouldn't say that's an impossibility.
What are we looking at?
Well, we're looking at sea cucumber right now.
Okay.
And we collected one very similar to this on the last leg.
How do baby sea cucumbers look?
I don't know.
I don't know much about the reproduction.
Hmm.
All right, I think we're good with that. Thank you.
I never saw it.
And then we're also looking at this scar over here just on the right edge of the frame. And sometimes you end up, we see these scars, sometimes they're much deeper and much bigger. And one of the leading hypotheses for their formation is they're actually beaked whales coming down and feeding or bumping into the sediment.
All right, well, we've kind of reached waypoint 2. You ready to start heading to the next one?
Yep. There's nothing magic about waypoint 2.
All right. 220.
Roger.
Bridge nav. Can we do 40 meters, 220?
Different type of sea cucumber here. These are the swimmy kind.
Push it there, Amber.
The
Come down, land, get a belly,
Elpididae,
get
I
a
think.
get a belly full of sediment, and then jump back into the water column and spend most of their time in the water column.
Like translucent?
Yep.
Wow.
So that all of that's its digestive tract.
Oh, that's cool.
And it has really cool tube feet. Here, watch it feed here as it sticks its tube feet out, grabs basically a handful of sediment and puts it in its mouth. You can actually see where it had a gap in feeding You see that it's got dense sediment, then there's a gap, and then there's more sediment lower in its digestive tract. So you can
Let's keep an eye out for those troughs in the
all
sediment.
right, science is happy, thank you.
All right.
Because I'm finding some pictures that look suspiciously similar. That might be beaked whales feeding traces. That would be really cool.
But no one's actually ever seen seen the whale down here, so it's kind of a question mark still in my mind. I don't have a better explanation, but
eDNA people, this is where you come in. I don't know if we have the primers, but
I mean, they're using CO1, so
yep.
it should show up. Here comes another one.
Ooh, yeah.
Yep. I would tend to call that a beaked whale feeding trace, for lack of a better explanation.
Very cool.
That is so cool.
Yep, matches up with the dimensions in this paper I'm looking at here.
And that's from the Mediterranean, right?
Yep.
Yeah. Beaked whales are a much lesser known group of cetaceans. They're pretty speciose as marine mammals go. There's a lot of different species, and many of them are super deep divers, and they're exceedingly hard to study because they spend so little time in the surface water. They tend to not breach. They tend to not, they tend to be pretty shy. We know very little about them. If I remember correctly, there's one or two species that are only known from just a handful of skeletons that have been washed ashore. And they're
Back on shore, we went to a film festival at Pickrick, the Palau International Coral Reef Center, and there was a film about some whale biologists who were surveying the National Marine Sanctuary. I'm trying to figure out if they saw any beaked whales.
They did.
They did indeed.
Yeah. I don't remember how many species, but I know they did see some.
Beaked whale. Longman's beaked whale.
Right. Push in there for a simmer.
Here's a sea pen. Looks like Colobelemnon to me. All right, thank you very much.
Bridge nav. Let's do another 50 meters. $50 more.
You almost gotta wonder if it's not actually the beaked whale doing it, but potentially whatever the beaked whale is chasing would actually in some ways make more sense to me.
Maybe. Do you think they're chasing fish and shrimp or going for things in the sediment?
I think they're probably chasing squid. which is what's kind of confused me about, like, I don't quite figure out what they'd be getting out of the sediment there in the shallow like that. But if they were chasing something and it was trying to elude them and impacted the sediment, you'd get that kind of glancing scar shape.
I think we're kind of flattened out there, Jaden. You could probably come up to, just come up a couple meters.
Copy.
That's good.
Oh, here comes another one of those sediment mounds that that I'm baffled by. Saw 2 of these at least on the last leg, and I'm just surprised by their scale. Like, there are plenty of things that live in the sediment that pile up sediment. This is just really big.
Looks like the gopher mounds in my yard.
Yeah.
It's got bed forms on top of it.
Yeah, we dug around in one of these last time, push-to-push core in it, whatever, and learned nothing other than it's pretty well mixed and something made it. But I can't tell you what. This one looks older. This one doesn't, because it's got the ripple marks and it's a little more, the sediment appears to be a little more worked over in it, and it's not as homogeneous, so I'm assuming this is an old one.
All right, thanks.
Deep sea sediment is actually pretty biodiverse, extremely biodiverse, given the relatively low energy, low food environment that you think about the deep sea. It's one of kind of the fundamental conundrums in deep sea biology or deep sea ecology is why is the sediment so diverse? So when I say that, I mean all the things living in the sediment. There's all these microinvertebrates, small things, all living in the sediment. And it's exceedingly diverse and extremely patchy. And it's a real question mark of why you can have biodiversity rivaling like coral reefs and tropical rainforest in some areas of deep sea sediment if you're willing to do all, study everything under the microscope. And it changes dramatically. You can sit there and characterize everything in one patch of sediment and then move just a matter of meters away in a different direction and find a very different community. And why it's so hyper-localized and so diverse is still a big question mark.
Bridge nav, another step.
Always amazes me for so much we know about down here. There's so much more we don't know.
Yep.
And that's why we're here, doing what we do, exploring.
Wandering around with a flashlight in the middle of a giant parking lot.
Nah, not even a giant parking lot. Trying to characterize the entirety of Mount Everest by walking up at one place with a flashlight.
Pretty much.
with a map generated by a photograph taken from a 747 flying overhead.
I really like that part of the analogy. Push in there for a sample.
Yeah. Let's get the sea cucumber and the brittle star, please.
Roger. That's good, thanks. Lone brittle star.
And Martina in the chat's also pointing out that there's other sedimented mysteries of different patterns of holes and stuff like that that we really don't know what causes them. There's a excuse me, a hole pattern called polydictyon that's been documented like 100 million years ago in fossil record, and we still see the exact same hole pattern with the exact same measurements in the Atlantic, and to my knowledge no one has caught the organism that makes it yet, despite a couple efforts to do so. And we've seen another linear pattern that Mike Vecchione has documented in the Atlantic. Fish.
Zoom fish.
Roger, push in about halfway for us.
This looks like maybe Acanthomus. See you later.
Giant polywog to meet.
Yeah.
Oh.
This is the bony-eared ashfish, right?
Yep, the unfortunately named bony-eared assfish.
Completely dusted, sorry. Maybe come up just a few meters, Jaden. I got kind of close to you there.
Okay.
All right, for our viewers, we are at 2,107 meters. We just passed waypoint 2, and we are coming to the end of our shift. So just be expecting a new crew to be coming in shortly. So I'll say thank you, and we will see you soon.
Enjoy the rest of the dive. We'll be back in 8 hours.
See you later.
Coconut. I'm pushing on the coconut real quick. You're still there?
Bridge nav, hold position.
Yeah, that was worth it. Yeah, I did a couple times too. Like it's worth sort of like just going and. Okay,
Oh.
you're muted there, Dan.
Take 2, let me know when you want to get back on the road.
Yeah, let's check in with Jeff and the back row folks and
Okay.
see how everybody's doing back there with their handover.
Yeah, I think we are ready to start moving up slope.
All right, head to waypoint 3.
Copy that. What we have for heading here? 215. Right. Bridge nav. We move 30 meters to bearing 215.
I just wanted to check in. Is everyone good for introductions?
Yeah, I'm good to go.
Okay, awesome. So
That's correct, thank you.
So to our viewers, hello. We are currently exploring an unnamed seamount west of Babeldaob Island, just outside the boundaries of the Palau National Marine Sanctuary. My name is Amanda Dedicatoria, and I'm sitting here in the Science Communication Fellow Chair. And we started with the back row yesterday, but I'd love for the front row to start off the introduction today. Max, are you good to go?
Yeah, I can go.
Yeah, awesome,
So,
thank you.
my name is Max. I'm sitting in the video engineer seat, and I'm a video engineering intern on Nautilus. That's all there is to it.
Everybody, Josh Chernov, Hercules pilot and ROV operations manager for OET.
Hey folks, I'm Dan Torriello. I'll be your navigator today.
Awesome, thank you. And Noelle?
Hi everyone, my name is Noelle. I'm the data logger for this watch, and I am the science coordinator for OET.
Hello, I'm Greta Sartori. I'm a guest scientist from the Palau International Coral Reef Center.
Ali, my name is Jeff Drazen. I am the watch lead for this watch. I'm a professor in the oceanography department at University of Hawaii, and I'm a deep sea ecologist. I specialize in food webs and deep sea fishes.
Good morning, everyone from Palau. My name is Menzel Ngirmeril. I work for the Office of the President here in Palau, but for this voyage, I'll be the cultural liaison on board.
Awesome, thank you so much. I really wanted to start off this morning with like a joke about how seasick I was yesterday, and Jeff was like witness to the motion of the ocean that was within me. But I would honestly like to invite you all to think of, as we traverse the waters of Palau, to think about our sister islands in the Marianas, including my home of Guam, as well as Tinian, Rota, and Saipan, as they experience Typhoon Man-yi today. I mean, I hope this serves as a reminder that it is an honor and a privilege to do this work, and that as the ocean connects these islands to us, we are also very connected to each other. So thank you, and I hope we see what we can explore for today.
So just a little tether maintenance there quickly.
All right, well, for those of you who are just joining us, we are exploring a seamount, which is to the west of Babeldaub in Palau's, just outside of Palau's National Marine Sanctuary. And we started this dive just a little deeper than here. And we are making our way up what looks like a very conical shaped seamount that's not too dissimilar in its geomorphology from the seamount that we dove on yesterday. And the top of this is quite a bit deeper, though, than the one from yesterday. We will probably top out at something like 1,150 meters or so.
Yep.
And the goal of this dive is to survey the habitats and the fauna in Palau's waters. This hopefully will provide some really novel insight into what lives here and at what depths and in what kinds of microhabitats, and enable us to have a better understanding of what lives in the exclusive economic zone of Palau overall, including in the sanctuary.
Gets a little tricky when you want to stop, because then everything starts to swing. So if we really want to stop and stay in that spot, you're going to have to stop the ship. So usually what the best thing to do is just say hold position, and then once they kind of hold position, then give them the thing to back up. So you're giving them like only one ask at a time without like stacking them up. They get a little confused. So stop the ship, give them a bit, and then ask for the next move back.
We haven't had this much sand since we were diving on the slope of the trench. Oh, there's a great sea cucumber over there. This is Benthodites. These guys get very large.
Can you zoom in, please?
How large?
2 feet long.
Wow.
But they have this apron around the outside of them, and these little cirri that stick up in pairs from their back. A number of other of these sea cucumbers also have those cirri, but there'll be more of them or there'll be less of them, and seeing just a few pairs like this and that apron around, that kind of skirt around them, sort of helps identify it as being in this group.
Is that a hexacoral?
Hey,
Where was that?
No, I was joking because it has 6 tentacles.
Jeff, do we know why they have this cirri? If it is any sort of adaptation?
I don't actually know. There's a fish on the right. I do not know exactly all the functions of those cirri. I presume that they are...
We'll zoom in, please.
Possibly sensory, but I don't know exactly what types of structures they would have out there.
I see.
Do we have any more Zoom?
Yes, we do, but
Okay.
Let's,
Gotta wait.
Just give me a sec.
Yeah, no worries.
And let's go for it.
Okay, this is a little cuskeel called Basocetus. Great. You can
Okay.
see, yep. Let's keep going.
Come wide, please.
We've seen a lot of those little cuskels, but they're usually below about 1,500 meters or so. Right now we're at about 2,070 meters, so that makes sense.
Maybe let's do you want to do a DBL reset? Seems like a little bit.
So we just got a listener
Yeah.
question specifically for Jeff. They're saying, they're asking, what fish should I get tattooed on next? Like,
Well, I